are going to learn about lactic fermentations in food and dairy products. As you know, fermented food and dairy products have been used in different civilizations since ancient time, and all these fermentations are primarily brought about by the action of a group of bacteria which are collectively known as lactic acid bacteria. These can be abbreviated as LAB. They are useful in fermentation of a diverse category of food product which include dairy, meat, fish, vegetables, fruits and beverages. These food and dairy products are highly popular due to many attributes which are useful for human consumption. First, they are convenient. Second, they are highly nutritive and stable, and they also have numerous health benefits. Let us learn about the important features of lactic acid bacteria. Lactic acid bacteria are gram-positive, fastidious in nature, they are acid tolerant, and in terms of their morphology, they are non-motile, they do not sporulate, they are either rod or cocci in their morphology. They are found in different habitats like gastrointestinal tract of animals. They are also found in dairy product, seafood product, soil and plant surfaces. Classification of lactic acid bacteria based on their fermentative mechanism is as homofermentative and as heterofermentative. Homofermentative include Lactococcus, Pediococcus, Streptococcus and Enterococcus. Their important feature is that they produce only lactic acid following the fermentation of sugars or other carbohydrate sources. The second group is heterofermentative. They are mainly Leuconostoc and Weissella. The important feature is that they ferment the glucose or other carbohydrate sources into lactic acid, alcohol, carbon dioxide, and this process is less energy efficient process. As mentioned that there are two groups of lactic acid bacteria, homolactic acid bacteria and heterolactic acid bacteria. Let us have a look of the pathway by which the carbohydrate or sugars are converted into the end product. First, we will look at the homolactic pathway. In homolactic pathway, the glucose is converted first to glucose 6-phosphate, which is converted to fructose 6-phosphate and fructose 1,6-diphosphate. This fructose 1,6-diphosphate is cleaved into two parts, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The glyceraldehyde 3-phosphate yields 2-pyruvate, which in turn will give 2 molecules of lactate or lactic acid. Now, let us see what is the fate of the sugars in the heterolactic fermentations. In heterolactic fermentation, the glucose is also converted into glucose 6-phosphate, 6-phosphogluconate, and then it is converted to ribulose 5-phosphate, and ribulose 5-phosphate yields xylulose 5-phosphate, which in turn is broken into glyceraldehyde 3-phosphate and acetyl phosphate respectively. The glyceraldehyde 3-phosphate molecule yield one pyruvate molecule which yields one lactate or one lactic acid molecule. This is in contrast to the homolactic fermentation where two pyruvates are formed which yield 2 lactate. So, here the lactic acid produced is half the value which is produced in the homolactic fermentation. The acetaldehyde component yields ethanol which in turn can also give rise to carbon dioxide. To sum up, we can say that the fate of glucose is the conversion of 2 lactic acid molecule in case of homolactics, whereas in case of heterolactics the glucose molecule is yielding 1 molecule of lactate and also 1 molecule of alcohol and carbon dioxide. Among the two processes, homolactic acid yield 2 ATP molecule which can be utilized by the lactic acid bacteria, whereas in case of heterolactic acid fermentation only 1 molecule of ATP is produced which can be utilized by the organisms. Some of the commonly employed genera of lactic acid bacteria are listed. They are Lactococcus, Leuconostoc, Pediococcus. These are all cocci in shape, and Lactococcus and Pediococcus are homofermentative, whereas Leuconostoc is heterofermentative organism. There are also rods like in case of Lactobacillus, which can be either homofermentative or heterofermentative. The other commonly employed lactic acid bacteria which are active as native fermentative organisms or as component of the starter cultures are Streptococci, Corynebacterium, Enterococcus, Aerococcus, Tetragenococcus and Eleucoccus. All these lactic acid bacteria are found in nature and some of these are efficient and has been formulated as efficient starter culture to bring about lactic acid fermentation of various food and dairy products. Lactic acid bacteria, as the name indicate, refers to their ability to produce lactic acid, and as a consequence there is a fall in a pH of the product. Then they also bring about proteolysis and they are also responsible for various other enzymatic reactions. For example, lactic acid bacteria, they bring about whey expulsion in case of dairy product. Second, they also curdle the milk and they are affected by the water activity. They are responsible for calcium retention in the dairy products, and also the milk protein lactose is converted to lactate, and their activity also inhibits the growth of undesirable microflora which is responsible for the spoilage of dairy products. Lactic acid bacteria, they also bring about the production of flavor, aroma, and they bring about the proteolysis, peptidolysis and generation of bitter peptides also takes place. Lactic acid bacteria convert the lactose sugar which is present in the milk to lactic acid, and there are other certain byproducts also formed which include diacetyl N-acetaldehyde, and these two are the major contributor to the flavor of the cultured milks and fresh cheeses. Now let us see what are the different type of dairy products produced by the metabolic activity of lactic acid bacteria. Lactic acid bacteria are the key organism for the production of cheese, yogurt, buttermilk, cottage cheese, sour creams and cultured butter. The different steps in the production of yogurt involves first the pasteurization of milk at 80 to 85 degrees C for a period of 30 minutes, followed by its rapid cooling to 40 to 45 degrees C. At this temperature, the milk is inoculated with 2 to 3% active starter culture. The starter culture used should be a combination of efficient microorganisms, primarily of lactic acid bacteria, and to ensure a uniform distribution of the starter culture, it is advised to stir the solution for a period of 3 to 5 minutes. After the starter culture has been uniformly distributed in the milk, the product is incubated for 2 to 3 hours duration at a temperature of 40 to 45 degrees C. The other option is to incubate it for 12 hours at 30 degrees C. After incubation, a coagulum is formed and it is advised to check the nature of coagulum. It should be a compact coagulum. At this stage, the acidity expected should be in the range of 0.8 to 0.9% lactic acid, and the pH value should be in the range of 4.3 to 4.2. The coagulum is refrigerated at this stage for 12 hours to prevent further decrease in the pH of the yogurt, and in order to improve the consistency, viscosity and flavor development. The product, the curd, is homogenized to a smooth consistency and then it is packed as a finished product. The lactic acid bacteria are also utilized for fermentation of a wide variety of vegetables and fruits. Fermentation of vegetables is mediated by certain lactic acid bacteria such as Leuconostoc mesenteroides, Lactobacillus brevis, Pediococcus acidilactici, Pediococcus pentosius and Lactobacillus plantarum. Leuconostoc mesenteroid is active during the early stages of fermentation and it results in a drop of pH to 4.6 to 4.9, and then the conditions become conducive for the action of more acid tolerant lactic acid bacteria. Pedococci and lactobacilli act upon at this stage. Lactobacillus is also used in the production of certain products like sauerkraut, silage and pickles. Depending on the use of salt, the lactic acid fermented products are grouped as dry salted. Dry salted means the dry salt is used for the treatment. Example include sauerkraut. The second group is brined, where only brine is used, for example in case of pickled cucumbers. Lactobacilli are used in the production of sauerkraut, silage and pickles. There are others which are referred as non-salted. Non-salted means no salt is added during the fermentation. Example include Gundruk and Sinki. Lactic fermentation process is also used for various type of meat and fish products. Lactobacillus species are used as a starter culture for the fermentation of meat to sausages. Lactobacillus plantarum is used as a starter culture for the manufacture of summer sausages, pepperoni and salami. Fermentation of fish and shrimp, for example, bagung, fish and negari, is also mediated by the action of lactic acid bacteria. Now, let us see how the lactic acid bacteria mediated fermentation inhibits the spoilage of the food product. There are certain attributes created by the lactic acid bacteria such as low pH, production of organic acid, production of bacteriocin and production of hydrogen peroxide and alcohol, which is primarily responsible for the microbial inhibition of the spoilage organism. The other parameters include the conditions like nutrient depletion, low redox potential created by the action of these organisms. These also contribute to the reduction and inhibition of the spoilage microorganisms. Lactic acid bacteria are beneficial in terms of inhibiting the enteric pathogens. They are known to possess the functional properties and are proven for their anti-cancer activity. They are also known to stimulate the immune system. Lactic acid bacteria are also found to reduce the cholesterol level and are known for their hypocholestemic action. Lactic acid bacteria also possess bacteriocin property. They are known to produce 4 different classes of bacteriocin, namely class 1, class 2, class 3 and class 4. These bacteriocins are responsible for inhibiting the pathogenic organisms. The fermented foods produced by the action of lactic acid bacteria are endowed with numerous functional benefits and they are recommended for good health of human being. There is a term probiotic. This term was given by Elie Metchnikoff and this refers to a group of bacteria that colonize the human gastrointestinal tract and they resist the low pH value and the bile salt. They prevent the growth of pathogens with their antimicrobial activity and they are considered safe for consumption and they also show no antibiotic resistance and therefore they are suitable for commercialization. So, let us sum up. Lactic fermented food and dairy product, they have been used in different civilizations since ancient time. Examples of lactic acid fermented food are ripened cheese, fermented sausages, sauerkraut and pickle, etc. It is one of the easiest way to preserve the food, improve its aroma and flavor. Lactic acid bacteria are the chief organisms involved in the fermentation of food and dairy product. Nowadays, starter cultures are employed which have proven efficiency, consistent performance and they are able to convert most of the lactose or other sugars to lactic acid and their other metabolites like exopolysaccharide, bacteriocins, vitamins are also of great importance for human nutrition. Diacetyl and acetaldehyde produced as a result of lactic acid bacteria contribute to the flavor of the cultured milk and fresh cheese. Lactobacillus species is often used as a starter culture for fermentation of a variety of meat, fish, and they are used commercially for the manufacture of summer sausages, pepperoni and salami. Nowadays, functional benefits of the fermented food products employing lactic acid bacteria is termed as probiotics. Positive effects of probiotic on human health include lactose intolerance, cancer, allergies, hepatic diseases, Helicobacter pylori infections, urinary tract infections, hyperlipidemia and assimilation of cholesterol. Therefore, lactic acid bacteria are useful not only in preservation of variety of food products, both dairy as well as non-dairy, but are also important as probiotic agent for the improvement of human health. Thank you.